Oh, okay. The only thing that I can tell you today is this is hot stuff. Hot stuff. We're in chapter 17. We're going from gene to protein. So I'm going to lay out our plan for this exciting week. So here's the big picture. We're going to kind of put ourselves here. Suppose this is all cytoplasm. Can't even see the cell membrane. Let's draw a little bit of nuclear envelope. Here it comes. Get some nice pores here. Cartoonistically. Okay, very nice. Looking good. Looking good. And looking good. Okay, so obviously cytoplasm. This would be the nucleus. And then living in here is our DNA. And let's give DNA some love. Throw some nice color on it. So remember, it's double-stranded helix. That's beautiful. And we're going to see a wonderful process here in the nucleus called transcription. That's our first stop, is transcription. Oh my, do we have a lot of good things to say. And then we're going to produce from that. We're going to produce from that this bit of stuff that's going to look like this. And I'm going to throw some colors in here. We'll see this later. Going to get this crazy business here. We're going to call this pre-messenger RNA. And then we're going to do something like this to do some processing. So I guess I'll make this like this. And this will be our actual mRNA. And it's this bit that goes out into the cytoplasm through the pores. DNA is held captive inside the nucleus. It can't leave. But the messenger RNA can. The messenger RNA is single-stranded, as is the pre-mRNA. The DNA is double-stranded. And then it's going to go out here to our friend that you might remember. As a matter of fact, I'll throw some on here. You know, you remember that there's a big system of the endomembrane system, and it folds out into endoplasmic reticulum. And some of this endoplasmic reticulum is going to be studded, bejeweled with beautiful little things called ribosomes, which are made out of, so there's a bit of rough ER, and these things are made out of RNA. One of the other kinds of RNA, which is called ribosomal RNA. And what's going to happen is that messenger is going to basically flow through here, and we're going to get protein. So if we do a small blow up right here, we would see that here is our membrane. Here is our ribosome. And then our ribosomal RNA is chunking through this thing. And as it chunks through this thing, as a matter of fact, I'll have it go that way. And then we're going to be building an extraordinary, beautiful kind of growing chain of amino acids. This is our polypeptide forming. Trying to put some different colored ones in there to kind of show what's up. This is what we're going to see happen. Look at that. So, just to let you know, in this little box, I should probably make it into a box. Let's see. I'm going to take that bit of membrane out. This is the box. Now we're going to see a lot more detail on this later. But this is the box that's right here. So this is the ribosome right here. This is the growing polypeptide chain right here. And then this, of course, is our messenger RNA, which came from the nucleus and now is going through here in units of 3. Every unit of 3 codes for a specific amino acid. We're going to learn all of that this week. We call this process here, we call translation. And then, of course, you know that the title says from gene to protein. Hey, you guys know that this amino acid sequence here is the primary structure of a protein, so it's going to get folded secondary structure, tertiary structure, possible quaternary structure. Look at that. That is our big, beautiful picture for this week. It's really exciting. Now, one thing I want to tell you is we're going to turn it up a little bit for some of the detail. You've heard me kind of criticize the detail at this part of this text. There's so much information you can get lost in the detail. I want to fold in a little bit of detail here just so you can bow to the greatness of this system and bow to the greatness of how much we know. I think if you get a little bit of detail at this point, you can actually understand the significance of DNA in our world today. I mean, you know the significance. I challenge you to commit a crime and get away with it these days because it's very difficult to not leave your DNA somewhere. And if you leave your DNA somewhere, they're going to get on it, they're going to sequence it, and they're going to incriminate you as the criminal. Wow. So much. If you think about our medicine today, if you think about our coronavirus today, the first thing that had to happen was it had to be sequenced. Way back at the beginning of this year, sequenced in China. That defines this beast, this bastard virus that has changed our world. And it's all about the glorious details of this. Now, of course, the virus itself, just to let you know, put it on the map, the virus is basically a piece of messenger RNA wrapped up in some proteins and some lipids. Let me say that again. This coronavirus is basically a piece of RNA that is wrapped up in proteins and lipids. That's what viruses are. And that virus RNA is basically a piece of messenger RNA that actually does this in our cells. It commandeers our cells, our ribosomes, to make its proteins, and it's a nasty thing. Anyway, that is why we are going to turn up the volume a little bit on some detail to get an appreciation for the true fundamental phenomenon that we call molecular biology. It's a whole field. Jobs, careers, money, molecular biology. Okay, so let's get it. So let's introduce the players for this a little bit. We're going to look at RNA now. And we're going to see three kinds of, we'll look at RNA. We need enzymes. We'll look at the three kinds here in a minute. And the enzymes are going to be what we call RNA polymerases. And we've got three of them. So the first one is that we have RNA poly 1. RNA poly 1 transcribes what we call ribosomal RNA. So that means I'm going to have to talk a little bit about this. This R means ribosomal RNA. And this is what makes ribosomes that stud the endoplasmic reticulum in eukaryotic cells, but are also floating freely in, we talked about these. We had the matrix ribosomes that we discussed. We talked about ribosomes in bacteria, so prokaryotic. We had ribosomes in chloroplasts. These things are floating freely. So ribosomes are big, big, big. Then we're going to have RNA poly 2. And RNA poly 2 is the beautiful one here that we call messenger RNA. And so RNA Poly 2 transcribes, that's the process, messenger RNA. And let's get our messenger RNA. And then our last one is going to be RNA poly 3, and poly 3 transcribes what we call tRNA, which we're going to name as transfer RNA. When it's all said and done, you'll know all three of these, what they do, and how they play into this beautiful process from gene to protein. Okay, what we're really going to have to do, though, where most of our detail is going to come in, is going to be right here. We are going to spend most of our detail crunching time getting into this crazy world of messenger RNA. It's a good board, it's a short board, but we need a big board. So I'm going to have to erase this one. Okay. All right. So what we're going to do is we're going to look at this whole process of making messenger RNA. And we are going to only focus on eukaryotes. You know, that picture I showed you at the beginning was obviously a eukaryote. There's a nuclear envelope. Boing. You know, what's that? Yeah, that's a feature of a eukaryote. That's the definition of a eukaryote. Truly nucleated. So we're focusing on eukaryotes, staying away from prokaryotes, because we're interested in eukaryotes. Making messenger RNA, and therefore we're going to, as I mentioned, focus on eukaryotes. So here's what we have to do. We're going to break this process into three kind of big overarching steps. We're going to see, first off, initiation. Second off, we're going to see elongation. Third, we're going to see termination. All right, those are our three things. So here we go. Let's start right here with initiation. We're going to create a new and pretty straightforward, simple kind of map approach, or kind of a cartoon of a chromosome, of a bit of double-stranded DNA. So here's going to be in our DNA right here. And right here, this is one of our new systems. So this is DNA. DNA in yellow. And what we're going to see is 5' here, 3' here, and therefore the reverse of that this way. And we're going to see that there's different parts of this thing. This area right here, this area right here, and then this area right here. So this is double-stranded DNA. And of course, I don't think I need to point this out, but this space here, just for fun, if you did this, would be basically the hydrogen bonds that holds that together. This is anti-parallel double-stranded DNA. It's our new cartoon. And so everybody knows anti-parallel means 3' to 5' one way, and then the reverse of that. Complementary base pairing. So this would actually be a whole bunch of bases all the way through. And we're going to have this part here, and we're going to call this a tata. And the tata business has to do with thymine adenine. Therefore, if that's tata, then this part would be AT, because it would be complementary. And we're going to name this top strand here. This top strand we're going to name as non-template. And we're going to name this strand here as template. So we got those two things. To further shed light on what non-template is, the template is actually going to be the bit that has the sequence in units of 3 that's gonna code for the messenger RNA. This non-template, therefore, we're gonna call this thing equal to non-template equals the coding strand. And basically what this means is that this is the same as the messenger RNA that we make. It's going to be the same as the messenger RNA that we make, except it is DNA. So basically what I'm saying here is that this thing would look just like the future messenger RNA that we're going to make, except for the fact, actually the pre-messenger RNA, but it's going to have deoxyriboses instead of riboses, and it's going to have thymines instead of uracils. So thymines instead of uracils. Standard DNA versus RNA work that we did several times in this class a long time ago. So that's what that is. And now we have a kind of an understanding. The next big exciting thing for us to do is to identify this part right here. This is what we call our promoter. Boom. Huge, huge stuff. Promoter is going to be defined as a DNA sequence. This is a DNA sequence where RNA poly 2 will attach. So that's like a place, oh, okay, I'm RNA polymerase 2. I'm the enzyme that says, hey, let's make some messenger RNA. And I need a place to start the party. They're going to start the party right here. This is where RNA poly 2 is going to bind. To let you know, we're going to see this process with some other things. When it attaches here, we're also going to see that in eukaryotes, which is what we're dealing with, that it also, this part of attachment also involves what we call transcription factors. Transcription factors are proteins. And we're going to put a beautiful drawing together in the glory of Technicolor chalk. Chalk is great. It's better than white. Those whiteboards, you can't really shade, but with chalk you can. We're going to have a fun time. So here we go. That also is a great board, but we just need a lot of real estate to tell this story. So here we go. Get used to this as kind of a general cartoon for the way we'll project it. We might even simplify it more, but basically get used to that. All right, here we go. So now, everybody, we're right up here at our promoter. So here we were. I've just got to put this up at the top so we can see it. 3 prime, 3 prime, 5 prime. Boom, 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 boom. This is AT, ta-ta here. So that's what we had. And now we're going to basically focus right here, as I mentioned, in this place. We're going to go down here. And what we're going to do is we're going to add to this process RNA, I'm going to change that and make it colored. Keep it color-coded. So RNA poly 2, which is going to be this big, huge, awesome thing that we'll shade up a little bit. I'm going to need to get a smaller piece of orange. So we'll shade this up a little bit. This is going to be that. And then we're going to have transcription factors coming in. And those guys are going to be small, and we'll kind of shade those up like that. Okay, so that's what it's going to look like. And so here's what's going to happen. Now, we've got a lot of things going on. We're separating strands, breaking hydrogen bonds, we're untwisting, the kinds of things that we saw during the DNA replication process. Here's what the whole thing is going to look like for us. We're going to see a bubble. And we're going to see the reverse of that here. It's this bottom strand that's important to us. And we're going to actually see these transcription factors and bunches of them. So we're going to see transcription factor here. Try to shade this a little bit. That's a transcription factor. We're going to see one here. We're going to see one here. These are all beautiful transcription factors that have come to party. We're going to see here. Look at that. We're going to see one here. So these are very important factors. Boy, yeah. Take a class in molecular biology and you can start breaking that stuff down. I mean, the detail can be overwhelming, but I like it that we're stepping into the detail pool, but we're not going to go crazy. Matter of fact, I can't go crazy. I don't know this stuff. I've got colleagues that do. I know the whole organism. That's my thing. So, there we have this process. Then, we're going to get, what's that beautiful piece of red? We're going to actually start right here, and we're going to show the making of our RNA transcript from 5' to 3'. This is our young RNA transcript. Okay, so where does this whole awesome thing called RNA poly 1 come in? It's a beast. RNA poly 1 is kind of enveloping this entire thing. So I guess technically speaking, we could completely shade this thing because this is RNA poly 1. So we're trying to get some orange shading here to show RNA poly 1. Look, it's a beast. Excuse me, RNA poly 2. I kept saying RNA poly 1. So, sorry for that. This is RNA poly 2. That's what that thing is. Look at that beast. Oh my gosh, this is beautiful. Alright, so that's what's going on. And the whole thing is going to happen. We have now, I guess technically speaking, we are really in the process of elongation. Because if we took a look at this boat later on, so this whole thing of attaching these transcription factors in RNA poly 2, that's initiation. But we've already added some bases here. And therefore, because of that, we've already done some elongation. So somehow in here, this whole process of putting it together is equal to initiation. These things all gather together, and then in this process from here on down, we're really going to be dealing with elongation. And this whole boat is going to move along the lines. So let's say right here, now we're over here in this bubble by the time we get here. And so RNA poly, let's actually get our let's get our boat together. So again, same transcription factors we saw before. And then shade that up a wee bit. So this whole thing, you guys can clearly see, has been chunking along. It's a giant bead, a sled, kind of working its way along the rope, the double-stranded rope. And so by this time, what we're going to see is our transcript is going to look like this. So again, let's get our prime here. That's 3 prime. Let's make sure that we honor our prime designations here as well. And so then way over at this end, we actually have our 5 prime end. That is our elongating. So this bubble goes this way. I think I would like to give this a little bit more length because that's kind of how I've rendered it here. Sorry about that. So this whole big barge is actually kind of, and as it goes, we're increasing the length of this, and this thing just kind of sloughs off and out, right? This whole sled is moving across. Wow, wow, wow. That is an extraordinary process. So that's elongation. What we have to do on our next board is take a look at, briefly, at termination. But let's go ahead and put brackets here to indicate what this is. Boom. Initiation, elongation, and look how beautiful and big that this is growing. So this is our developing RNA transcript. So I'm going to put that there. Good grief, that is beautiful. Absolutely beautiful. I mean, so beautiful I kind of hate to erase it, but I guess we'll do it. Into some termination. So on termination, basically what I want to say is that we're going to see detachment. Detachment of RNA. Let's make that orange. That's what we've been doing. Of course, and factors. So we got that, and that's the end of the story. But what we have to do here is get into the beautiful of this process. So after this thing is over, now we have an mRNA. Well, we have an RNA transcript. Now this RNA transcript has a big future. What's got to happen to this RNA transcript is that it's going to be modified. And it'll be modified by a process called RNA, a set of processes called RNA processing. The processes of modification are called RNA processing. And there's going to be two big parts to RNA processing. But what we need to do is draw the whole map. As a matter of fact, let's put the parts. The first one is going to be, we're going to have alteration of the ends. And in the alteration of the ends, this is Roman numeral 1, sorry. In the alternation of the ends, we're going to see first off the making of a 5' cap is added. This 5' cap is going to be made of a methylated GTP. And you're familiar with the whole TP process, triphosphate, and this particular one okay, so here's some selected figures from the text. These are pretty cool figures. I think it's good for us to take a look at these. A lot of what I'm drawing is actually my rendition of these figures. So, this first one, figure 17.9 on page 343, this one shows this kind of double-stranded DNA kind of cartoon model, and then the initiation phenomenon. Beautiful. So, just to point out some parts then, here's our first model right here of DNA. And remember that we've got the non-template strand and the template strand, anti-parallel, always identifying the primes. 3' to 5' for the non-template, and then reverse of that for the template. Then here's our exciting region here. There's the Tata box. And of course up here it says Tata, T-A-T-A. And then here, the complementary version is A-T-A-T. They call this whole area the promoter. And remember that that promoter was essential in eukaryotic kind of transcription. So we have eukaryotic promoter, and then we have transcription factors to add. Now, I've actually eventually drawn all of them, but here they're showing up, and they're arriving. And then here comes RNA polymerase 3, gloms on to this big kind of double-stranded process. It's a massive, beautiful thing. And of course, now we've already started our elongation process because RNA transcription is occurring. This whole unit, transcription factors and RNA poly 2 and the separation bubble that forms is just going to move. And that's going to get red, that red RNA transcript is going to get longer and longer and longer. So that is the textbook version of what I've already shown you. I think we did a fine job there. So here comes the next one. So this actually is the previous figure, 17.8 on page 343. But this one shows all the processes. We've got initiation, elongation, and termination. So here we have the promoter. It's stuck there. We've got a lot of room to travel. This is going to be the transcribing unit. We'll see more on this in a minute, by the way. And then we're going to go into this initiation process. Now notice that to probably make it less complicated, they haven't shown the transcription factors here. There's no little purple spheres on this. This is just the whole big guy, the whole big RNA poly 2. There's a very tiny little transcript forming. That's the thing. Now, we're looking at this in the same frame, but later. Later the thing has moved a lot further, and now the transcript is very beautiful and very long, and it continues. And I think that the idea of transcription is that we're growing. The first part made is at the 5' end, and the last part is at the 3' end. So 5 is above 3. It's a downhill process, so they're calling it downstream. It's going downstream. In the red is the process. Making of that is downstream. because of course it's upstream if you look at it as going up the DNA. But downstream is the concept in terms of the transcription, which is the synthesis of the messenger RNA. Downstream. We're adding at the 3' end, we're adding at the bottom. Okay. Then we go all the way through, and the termination process takes place. Dump off. This is our complete transcript. Remember that transcript, we got to do a lot to that transcript to kind of get it ready to be a true piece of messenger RNA that actually goes out into the cytoplasm. All right. And this particular one is a guanine triphosphate. How cool is that? It's the first time we've seen that. We have adenine, also known adenosine, and here's guanine. So this is a nucleotide with a couple of extra phosphates is what it is. That's a nucleotide. So you have a sugar, you have a phosphate, and you have the base. And then you have 2 extra phosphates here. So that's what it is. Methylated GTP. And then the second thing that we do is the other end. And this is going to be what we call a 3' tail. And that 3' tail that's added is actually made of poly A. So it's made of poly, which means many, A, which means adenine. So here we have a whole bunch of adenine nucleotides that are added, a whole bunch of A's at the end. So we've got that. Let's take a peek at this, perhaps. I think I've got enough room right here. So here we're going to put the whole thing together. And right here we'll have our 5' end. Come on. Jeez. 5 is the number. 5' end, 3' end. And we're going to have really good looking stuff right here. So let me look at this for a brief moment. Okay. Okay, got my blue chalk. Okay. Blue bits, red bits. Talk more about that in a minute. Blue bits and red bits. And let's see. Probably need to get a blue bit on each end. Okay, make sure you get that. It's beautiful. I'm going to actually solidify these so they look better. These colors definitely will mean something very, very soon. So we've got that going for us. Then we're going to have a whole area right here that's going to go, that's going to get a name. So this area is going to get some name. And then we have this area here, which I think will just make it look a little bit different. We have this area here, which looks like that. Okay, so that's the whole piece of RNA that got spit out from the process above. Except for the fact that we've added this So what we're going to see at this end is actually going to be, actually it's right here, this very tip. This particular part is going to be a GT, you can write it. This could be, matter of fact, let's take it out and do it. Here is P, 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 and G. Now, I know that's grotesquely large compared to what we've done, but that's what we've got right here. So, that's GTP right here. We've added that. This part right here, we're going to call a UTR. And the UTR is an untranslated region. So it's not going to be translated. That's what U, T, and R mean. We have another UTR over here, right there. So we have UTRs. And we got them over here, we got them over here. Uh-oh, I put that in the wrong place. Sorry about this. This is the UTR. Getting a little ahead of myself. And then finally, this bit right here. This is our poly A tail. Okay, so here's the deal. Now that we have the picture, this part got added. This was added. This part got added. And this modification thing added. Added. I hope that color stands out. This is added. This is our tail. Then this is our cap. And we identified these. This is a 5' cap. So obviously here's 5', and this is a 3'. This is a 3' tail. That is the whole beautiful thing. Now I'm about ready to completely knock your socks off, because this thing right here is the money. That's the exciting stuff. We're going to name that right now. This part here is going to be what we're going to call our protein coating segment. And the next board is going to be 100% about that protein coating segment and what we have to do to it. Technically speaking, from our first board today, this bit right here is technically speaking, all of this bit is pre-messenger RNA. That is a good board. Right? So here is our primary transcript. This is the whole big thing. After we've modified it, there were these different parts of modification. First thing we had to do is add a 5' cap, which is a guanine triphosphate. Pop, 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 right there. Then on the other end, we modified the ends, right? So we're going to do the tail, and that's a poly A tail. Look at the numbers of those. There's 50 to 250 adenine nucleotides here. Of course, remember, nucleotide means sugar, phosphate, and base. And the base in this case is adenine. So 50 to 250, all the way across. Here is actually the good stuff in the middle. I pointed out to you UTR, and the UTRs are untranslated regions. Got one here, got a big one here. I did not point out the polyadenylation signal that's in the middle of the UTR. You can read about that if you want. Can't get all these details in. Here's the money. We've got a green bar that's a start codon. More on that later. We're going to identify that. And then we have a stop codon here. There's only one of these. There's 4 possible of these. And then all of this stuff is going to be actually the protein coding segments. So the other part of this that we're going to see is that we've got things called introns and things called exons. And it's the EX, exon, expressed that make the code. So we're going to have to get rid of some of these parts and keep some of these parts. And that, of course, happens in splicing. So now it gets really fun because we're going to take a look at that special part. Here's what we've got then. Let's take a look at the original DNA. In that DNA, we could actually say, okay, so this is going to be kind of our gene region. We actually could say, let's kind of code this. And then let's put some of this in between, as we did before. And then let's take this thing right here and take a look at, so this is obviously DNA. And then let's take a look at what we get out of this. Here's what we're going to get out of this. We're going to get a piece of messenger RNA that's going to look like this. And that piece of messenger RNA is going to have 2 different colors, and these are going to make sense really soon. So that's what that's going to look like. Now we have to also identify here that this is 5 prime, this is 3 prime, and I think we should actually stick on this a cap, and that's a GTP cap, and then stick on this is the tail right here. So this is the poly A tail. This is this whole business, and what we're going to do is identify these bits. The blue stuff, blue is equal to what we call exons. Red is what we will call equal to introns. Now the crazy thing about this, I'm going to introduce it right now, is that exons have this EX bit. EX for expressed. And basically, therefore, these are actually going to be part of our code. That's going to be part of our code, and therefore, these parts are not part of the code. So what we're going to do with this process is quite remarkable. We're going to actually go from here, from this part right here, we're going to go through a process called splicing, and we're going to end up in the splicing with this. A short bit, because we've cut some stuff out that's going to be actually what we would call, let's get our 5 prime cap. We're going to end up with this part right here, which is actually going to be our true mRNA. This is the part that's going to leave the nucleus, as we saw before. This guy's leaving the nucleus. Leaves nucleus, and its destination of the great process of translation. Destination translation. Okay. That is awesome. I need to tell you a little bit more about this. More info coming soon on the next board. Okay. So here comes our next info. Splicing is what we're talking about. And I guess if we went back to our original outline, that we would have said that splicing gets Roman numeral number 2. So adding the cap and the tail was Roman numeral number 1 for modification of that pre-mRNA, and then number 2, Roman numeral 2, this is our modifications. Mods would be called splicing, and it occurs in the nucleus. And what's going to happen here is we're going to see these loops. So the blue bits are going to look like this. Cap, tail. The red bits are going to be loops. Oh my God, that's amazing. So these are our introns. And these introns, did I back my color up? Nope, I'm good. These are going to be introns, and the process is basically going to be intron loops, and they're going to be what we call, they are excised. So what has to happen here, you guys, is that we have to actually break the sugar phosphate backbone of this single-stranded bit of RNA and actually chunk these pink bits out and then seamlessly fuse all of these blue bits together. So that takes a little bit of doing. The whole thing that forms to do this is called the spliceosome. So there's stuff going on here. Whoops, let's see, spliceosome. And the spliceosome has some parts to it. What it has, this is our cap, tail. The spliceosome has some bits to it. It includes what we call S, N, R, N, Ps. And these are called small. It's supposed to be, small, nuclear, ribonucleo, protein particles. So that's what these guys are. That's what SNRNP stands for. And what these things are equal to would be RNA plus proteins. So these things are involved in the spliceosome, and then we're going to add to that in the spliceosome other proteins. And then we're going to add to that our primary transcript. And that's the whole thing, introns, exons, and all of this thing here. All of these bits right here added up equal what we call spliceosomes. That's a spliceosome. So let's take a look at what happens here in the general sense. So after this whole process, everything that's on the board here, after this splicing process, what we're going to get is this, which is our RNA, plus we're going to get bits of this. That's what we get out of the deal. And this is our mRNA right here. Are you with me? I hope so. So now we've got that. That's ready to leave the nucleus and go do what it does. Before we do that, you've got to hear this story. So I'm going to erase most of the board and leave splicing to show that we're still in the topic. This is what I would call a super wow thing. And what this super wow thing is what we call alternate splicing. So in alternate splicing, we have one gene. Now we have to remember that a gene is basically a sequence of DNA at a locus. And we know that the locus was a location on a chromosome. These are things that we learned most recently and most specifically during our Mendelian inheritance phenomenon. And when we were talking also about meiosis, we put all those things together because we had to understand concepts of independent loci, one's on different chromosomes versus linked loci, one's on the same chromosomes. So I hope that's ringing the bell. And this alternate splicing thing is super wow. Super wow. Here it goes. Alternate splicing says that one gene makes different, through splicing, different mRNAs. What? Even to wow it up a little bit more, in eukaryotes, well, in humans, there's about 35 to 59%, I have no idea where they came up with 59, 35 to 59% of human genes exhibit some form of this alternate splicing. The old story was one gene, one protein. The old story, one gene, one protein. The new story, one gene, several proteins, or at least more than one. Now, I told you that I'm the whole organism guy, but I'm a man of science that's, tries to keep a pretty broad toolkit. I'm not the guy holding the pipette in the molecular biology, but if you take a look on Blackboard, you're going to see a publication on my mosquitoes that are in the June. The lead author is one of my mentors named Borowski, and of course it has Hancock and others. And if you take a look at this, there is a paper on Culex mosquitoes, and we're using a splicing phenomenon. We're defining a splicing phenomenon that enables the mosquitoes to actually digest blood. And the splicing phenomenon is enabled by the process of sugar feeding, which is my expertise. So here's a mosquito that comes into the world, and if that mosquito takes a sugar meal, there's a splicing event that enables them to express polypeptide degrading enzymes called trypsin. So protein digesting enzymes. digestive enzymes. But it doesn't have those enzymes without this splicing phenomenon that's dependent on sugar feeding. Whoa, whoa, whoa. So that's super heavy, super cool. And that's why I think this thing sends just bumps down my spine. Super wow, this splicing phenomenon. It's delightfully complicating, isn't it? You know, it's kind of cool maybe to look through the world of genes and gene phenomena in simpler systems. The simplest being, of course, the craziness that happens in the damn viruses that are on our minds right now. But of course, fairly simple in prokaryotes. But man, when you get up into the wonderful world of eukaryotes, wow. It is extraordinary. So now, where we sit is that we have messenger RNA. That messenger RNA is ready to leave the nucleus in its eukaryotic cell and travel to the factory. The factory for putting polypeptides together is going to be ribosomes, and the ribosomes we only need one other thing. We need raw materials to make proteins, so we need amino acids. And those amino acids are going to be brought to the construction site, if you look at it that way, aka the ribosome, with transfer RNAs. So now we actually have 3 kinds of RNAs. We're going to spend a little bit of time breaking those down: mRNA, rRNA, tRNA, and then we can see the whole thing comes together, and it is extraordinary. So that's all I got right now. See you in a few.